So good morning, students. So today we will discuss these organs of the immune system. In the organs of immune system, we have two types, that is primary lymphoid organs and secondary lymphoid organs. In the primary lymphoid organs, we have bone marrow and thymus, and in the secondary lymphoid organs, we have lymph nodes and spleen and tonsils. So in this, here you can see that these stem cell precursors in the bone marrow. So this is the position where there is a mass of cells which have stem cells, and in the bone ends there is a bone marrow, and here all the processes of hematopoiesis will take place, and all the blood components will be produced. And also, in addition to blood components, we will also have this white blood cells, T lymphocytes, B lymphocytes. So every type of these cells which we have discussed in the hematopoiesis, they will take place in the bone marrow. Then the maturation of the B cells will take place in the bone marrow, and the maturation of the T cells will take place in the thymus. So we come to this thymus. In the thymus, we see that this thymus is a lymphoid organ overlaying the heart, seeded by lymphoid stem cells from the bone marrow, which differentiate into T cells. It is bilobed and organized into lobules separated by connective tissue, septa. So these are the septas. These are the structures, a section of this thymus. So each lobule contains a peripheral cortex and central medulla. Peripheral cortex and central medulla. So here you can see that it have special types of cells, that is these cells which have the cytoplasmic projections, and these are termed as dendritic cells. So these are very important for the functions of this thymus, because the thymus is for the maturation of the immune system, for the maturation of T cells. So, and T cells should be educated so that they should know what is self and what is non-self. So thymocytes are thymic lymphocytes. The repertoire of T cell antigen receptor develops, and the deletion of autoreactive cells occurs. Autoreactive cells means those cells which are reacting with the self antigens or self proteins. They have to be removed from the system. So it's a process of T cell selection, so that only those cells T cells survives that can recognize the non-self proteins, and if there are some cells which are reacting with the self proteins, they have to be removed from the system. They should die through process of apoptosis. So deletion of autoreactive cells occurs during T cell maturation in thymus by interaction with antigen-presenting cells. The process involves proliferation of immature cells, but most thymocytes die during selection by apoptosis. Means 92% will die, only 8% will be left. Thymic cortex, the outer zone of thymus, contains about 85% of the total thymocytes. These cells are immature, express CD1 in humans, and divide rapidly. Most cortical thymocytes expresses both CD4 and CD8 and are hence called double positive cells. So these thymus is more active in the young age, and its size will be about, its weight will be about 75 grams. But in the old age, its size, it will shrink and its size will become maybe 3 grams or 5 grams. So it shows that most of the immunity will develop in the young age. So therefore, we should immunize people, children at a proper time. And also if there is some exposure from natural sources, then the immunity will develop. The thymic medulla contains relatively few lymphocytes, but they are more mature than those in the cortex. So cortex have immature cells and thymic medulla have mature cells. Thymic epithelial cells are a network of antigen-presenting cells expressing MHC class II that extend throughout the cortex and medulla. So these MHC2 molecules are basically, they are presenting process antigens or epitopes to the T cells so that it will help in the selection and maturation of T cells. Basal corpuscles are walled structures of epithelial cells seen in the medulla of human thymus. Their function is uncertain. So, basal corpuscles. So you can see most of the activity will be in the cortical region, and these dendritic cells will help in the process of selection and maturation of T cells, and they will play an important role in the development of T cells. So next organ is lymph nodes. So lymph nodes in the lymphatic system, there are lymph nodes, and these lymph nodes have, you can see that they will have these cortical regions and paracortex and medulla, and they will have primary follicles, they will have secondary follicles. So these lymph nodes are encapsulated organs that punctuate the lymphoid network and contain aggregations of lymphocytes and antigen-presenting cells. They are strategically placed to intercept antigens from the periphery. There are large groups of lymph nodes in the axilla, groin, and neck. The mesenteric lymph nodes are large and well suited to protect the body from antigens and pathogens from the gut. So basically, there are two systems, that is lymphatic system and blood vascular system, and the antigens which will come in the lymphatic system, they will be trapped and they will be, immunity will be developed in the lymph nodes, and these antigens which will come through the blood vascular system, they will be trapped in the spleen, and then they will be educated and immunity will be developed. So spleen is an encapsulated secondary lymphoid organ that lies in the peritoneum, beneath the diaphragm and behind the stomach. It contains two types of tissue termed the red pulp and the white pulp. So it has these two types of tissues, red pulp and white pulp. Red pulp. So spleen lobules. So it is similar to the, you can say, lymph nodes and thymus, that it have these lobular structures. So these, so basically the red pulp consists of a network of splenic cords and venous sinuses lined by macrophages which affect the destruction of effete erythrocytes. Plasma cells may also be seen in this region. White pulp contains the majority of the lymphoid tissue distributed around the arterioles. So, marginal zone is the outer region of the pulse. It contains slowly recirculating B cells and CD149 marginal zone metallophils and macrophages which present T-independent antigens to B cells. So, spleen is also termed as graveyard of the red blood cells. So, the degradation of red blood cells will take place in the liver and the spleen. And also this spleen is helping to make this trap antigens which will come from the blood vascular system and will help to develop immunity against those pathogens. Another tissue which is termed as GALT or gut associated lymphoid tissue. In the gut, as the name indicates, that gut. In the gut, there are these tissues are follicular B cells areas and epithelial dome, M cells and T cell areas, and here these components or cells of the immune system are concentrated so that it can effectively help in the production of immunity against pathogens so that they will be trapped and the selection of T cells and B cells will take place and induction of immunity will take place. So, GALT. GALT comprises the mucosa associated lymphoid tissues of the gut. They include the focal accumulations of lymphocytes in the lamina propria and Peyer's patches, which contains high number of IgA producing B cells and plasma cells. T regulatory cells secreting IL-10 and TGF-beta are common in the lamina propria, helping to control immune response against dietary antigens. So these are the major primary and secondary organs which help our immune system to protect our body and educate the T cells and B cells so that a proper immune response will be produced. So the primary lymphoid organs are bone marrow and thymus. Secondary lymphoid organs are lymph nodes, spleen and gut associated lymphoid tissue or GALT.